concluding webinar of this series in Health and Diseases, Contemporary Concepts. Today we are having with us Professor Mikul Mehta, Professor of Neuroimaging and Psychopharmacology at King's College London. Mikul is the head of the Neuropharmacology Group and Section in the Department of Neuroimaging, Deputy Lead for the Neuroimaging Team of NIHR Morstan Biomedical Research Center for Mental Health, and Director of the IOPPN Center for Innovative Therapeutics. Mikul joined King's College London as a postdoctoral researcher in neuroimaging way back in 2003, with the aim of setting up a research group focused on neuropharmacological neuroimaging using MRI methodology. He worked with positron emission tomography at Imperial College for a number of years as an MRC training fellow before moving to King's College. A Wellcome Trust Value in People Award enabled him in this transition. His work was recognized by British Association of Psychopharmacology Young Investigators Award. At King's College, his group brought about quantitative methods to identify drug mechanisms and classify different compounds and used classic tracking methods to develop assays for drug modulation. Mikul has been involved in the British Association for Psychopharmacology in organizing the annual summer meeting, including a lecture for the public. He also co-leads a bi-year residential course in non-clinical psychopharmacology. In terms of public engagement, he and the group members regularly work with the scientific and general media on articles, interviews, and programming uh, as a social engagement. With this, I welcome Dr. Mikul. This webinar is organized in collaboration with Institute of Life Sciences Bhubaneswar, National Academy of Sciences India, Allahabad, and Indian Association of Neurosciences, and as a part of our Ravenshaw 150 Distinguished Lecture Series. I welcome Dr. Mikul to deliver his talk. Dr. Mikul.
Great. Thank you very much for that introduction, and thank you very much for the invite. I'm delighted to be talking to you, even though I can't see you all. I will share my. I think I am already sharing my screen, and it's on slideshow, so I can't see the app anymore. So if someone can tell me if this isn't working, that's fine. Otherwise, I will just continue. My introduction told you a lot about what I do in neuroimaging and psychopharmacology, but I also collaborate very closely in developmental psychology, and that's the focus of the talk today. So where am I based? So we are based in King's College London, in London, of course, and we have two campuses. So on the left-hand side, you can see our campus near Big Ben, near the centre of London. But we're not there, unfortunately. We're a bit further south, down just off the bottom of this map, over here in an area of South London called Denmark Hill. And it's a fantastic site because what we have on this site, on one side of the road, we have the Institute of Psychiatry, Psychology and Neuroscience. And you can't see really here, but these buildings are different colours. Some of these buildings are university buildings and some are the buildings of the psychiatric hospital. But you can see how well we are integrated. And I work here in the imaging centre. Across the road, we have the general hospital. We also run a scanner in the clinical research facility, and there are university buildings on the general hospital site as well. So we're a major medical centre in the south of London, and we're also the national centre for psychiatry as well. And we lead on neuroimaging in psychiatric disorders. So I'm going to tell you about this study we have been doing for many years now on the impact of institutional deprivation on subsequent brain structure and function. And this is a story that goes all the way back to the late 1980s, to the fall of the communist regime in Romania, headed up by Ceausescu. And they had very, very poor economics. They had real poverty, but they also had very poor access to contraception as well during this regime. And there are an excess of children born, and the children exceeded the number that families could really care for. So that created a lot of orphans, and many of the orphans were not biological orphans, but economic orphans, and they were held in institutions. And when the regime fell, there was shock around the world when the conditions in many of these orphanages were revealed. They were incredibly poor. I'll just highlight a few factors. First of all, the diet was severely restricted in these children, many of whom were infants in their first years of life. There were high rates of disease, and there was very little social and cognitive stimulation. And we already know from studies going back to the 60s, some of which were in monkeys, non-human primates, that lack of social and cognitive stimulation in those very early months and early years can have a profound effect on later brain function and development. There was also no personalized care, very little opportunity to form any selective attachments, arguably no opportunity to form selective attachments, all things that we think are very important in these early developmental stages. So the assumption is that normal development would be impaired. Okay, all the normal triggers that you'd expect at key moments in these early years were lacking. So the question is really why do we think that this could impact development? Okay, how it could impact development, and I'm going to give you an overview of what I'm going to talk about. So first of all, I'll just put them all up. Sorry. I'm going to talk a little bit about early experiences on brain development. I'm going to dwell a bit on why this study is important. I'll give you an overview of the findings, and I'll focus mainly on what's been happening in the brain, because that's been my area of focus and my role in this study. So hopefully bear with me. I've got a lot of text on the next few slides, but there's a few key ideas I want to get across. The first one is that we do know that there are many, many negative experiences that you can have as a young child or an infant. You can be maltreated, you can experience neglect. There's parental stress and conflict, there's abuse and trauma, there's being in a situation of conflict, and there's also all the stresses related to poverty as well. And many of these stresses can be physical, and all of these stresses we think have mental correlates as well. And I put institutionalized care in brackets here because this is what I'm going to talk about later on. But we do know that all of these forms of negative experience have been associated with later psychopathology. So that is increased risk for various disorders, mood disorders, and developmental disorders. And we do know that the brain circuits that are implicated to be vulnerable include areas such as the hippocampus, important for memory and everyday functioning, the amygdala, the important node in our emotional circuits, and frontal lobe circuits, which are really important for the high level functioning that we associate with everyday functioning as a human being, and getting on with society, getting on with the people around you. Okay, and there are some reviews down here. I'm very happy to share references if you want to find out more. But these studies are very difficult to do, and they're very difficult in terms of attributing causation. And why is that? Okay. Well, first of all, the sorts of disorders that we're talking about here, I'm going to focus really on, I'm going to highlight maybe autism and ADHD, really. These are developmental disorders that have repeatedly shown to have higher rates in those that have experienced some of these very early maltreatments. They also occur at higher rates in the institutionalized cohorts from Romania that I talked about, and I'll show you that data in a moment. So the heritability for, say, ADHD, it's about 0.8, and autism is about 0.6 to 0.8. So very, very similar. And heritability is not how likely you are to get the disorder. It's trying to capture phenotypic variation. So it's the extent to which a person's genetics predispose them to a condition, or how the but what it doesn't do to us actually is how the environmental causes of these genetic variations play out. Okay. And the model overall is a phenotypic model where genes and environment contribute to the phenotype. Okay. So the hereditary estimates that we have for these disorders depend on the type of experience that people have in a population, and typically they're in a normal environment. Okay, by a normal environment, I mean the normal society that we're in. I'm not talking about extreme environments like the Romanian orphanages. Okay. And the most common model used to describe heritability is the ACE model, and the reference to the ACE model is at the bottom of the screen. Okay, it's a classically used model, and it has three factors. The additive genetic factors, known as A, they're all the genes that might contribute or predispose you to certain psychopathology. Common environmental factors that you experience maybe with other people in your environment, typically siblings. And there's random environmental factors. Every model has to have a random factor, things that are really important that we can't really measure very easily and account for, that we might want to learn about. Okay. Now, what's important about this model is that the interaction term between these factors is thought to be quite low. It's a multivariate normative model of liability for disorders. Okay. And we know that there obviously are going to be issues with this type of model, and there are going to be correlations between some of these factors. But it's the most common model, and it works incredibly well, actually. There's plenty of data on twin samples demonstrating how this model works very well. And I won't go through that data today, so I can focus on our own study. Now, how does this link to our study? Well, what these models do say is very little about the potential of extraordinary environments to cause a disorder. And before I go through the text here, I'll give you an example which I quite like from Richard Lewontin about crop growth. And so what you've got here on the left, for example, is lovely crops growing nicely. And if we imagine they've got the perfect environment and every single plant here is exposed to exactly the same sunlight and water and soil quality, well, any variation between the plants, we would then be quite confident is genetic. Okay, so these plants are doing very well. If you imagine a situation like on the right, here you can see the crops are shorter, they're more stunted, they're not growing as well. So if we imagine this environment as a drier environment with poorer quality soil, but again, every plant is exposed to the same environment, the same poor quality soil, and the same arid conditions. Again, the phenotypic variation here between plants would be 100% genetic. So what you've got here is you've got the variation here is genetic and the variation here is genetic, but it's very clear that the environment has had a huge effect, but it's had effect on everything. Okay, so that's what we're thinking about. It's a very simple example. That's how we're thinking about these extreme environments that the Romanian orphans may have experienced. So genes are strongly implicated in normative development, but actually an extraordinary environment may provoke a disorder in an otherwise resilient brain or a strong brain. So the genes of these crops and these crops may be the same. So the potential here to be strong is very high, but the environment doesn't allow it to happen. Okay. But also, you know, the environment may help prevent or remediate disorder in a vulnerable brain. So if you're in a terrible environment, you know, you might have a great brain, but unfortunately you're in a bad environment. So just like this crop failure, there's going to be difficulties that you're going to have. Or you might be vulnerable, but you're in such an amazing environment, like here, that you can overcome those difficulties and you can grow well. Okay, so it's important to remember that extraordinary environments can work both ways. So hopefully this makes sense, and you can understand over these two slides why we feel that our study is very important, because it contributes to our understanding of these extraordinary environments. Okay. So the brain is highly neuroplastic. Now, neuroplasticity is fantastic, but it is a double-edged sword. So as I said before, just to repeat that with a picture, can adverse environments be so bad that an otherwise strong brain will not develop well? Okay, and the adverse environment here is the Romanian orphanages. So even though they might be adopted out of that environment, it's so adverse it affects brain development. But maybe an enriched environment is so good that it could help reverse or put these brains, the brains of these individuals, their own personal development back on track. Okay? So you've been in an enriched environment, and because the brain is so plastic, the brain can respond to different environments. Then if you take the brain out of this environment, the person out of this environment and their brain development, and put them in an enriched environment, maybe that can be beneficial. That's exactly what the Romanian study is. Just to emphasize that the value of this study, if you've got a poor environment, let's say parents, this example, you've got parents that create the adverse environment because of parental conflict. Maybe these particular parents have had developmental disorders themselves, and that is a contributor to the difficult environment of the child. Okay, well, that might obviously affect the environment, and the child might express the disorder as well. But is it this poor environment that is affecting the child's development? So here's the phenotype of the parent, okay, may contribute to their behavior, but the phenotype of the parent is also passed on to the child through their genes. And this might also directly influence the expression of a disorder in the child. So again, this just illustrates how we can't simply think about genetics and environment as separable phenomena, because they might have common cause as well. And the environment might have a big influence. It might override the genetic effects as well, as we might see in the Romanian adoptees. Okay. And there are other pathways in this model as well. Okay. So this particular study, I'll pull up these slides. I introduced the environment that these infants were in, a terrible environment, and they were adopted around the world, and this is the UK adoption we're looking at here, from age 1 to 43 months. And typically in the UK, a family which will adopt a child will usually be reasonably well off, have a house which allows them to have a room, and they're usually typically nurturing and supportive family who want to make a difference in a child's life. These are people who are really willing to put that extra effort in to create a warm and loving environment for this child. So, and we have interviewed these families, and these environments have been reviewed both by social services and as part of the research, and they really are amazing families. So these children, these infants, went into these families, and they've been in there for well over 20 years. It's approaching 30 years at the moment. Okay. So what's unique about this work is that the time at which they were adopted was a single point in time. At that point in time, we knew that this environment ended and this environment begun. This is a very unusual situation that you don't often see in studies of maltreatment. There are many studies of maltreatment where the maltreatment or abuse might occur within the family, and they might enter a slightly better environment but still have contact with this environment, or still be within the family environment with support, for example. And so it's not entirely clear what the window of the experience is. Here it is very clear. Okay. And because the adoption was based on who was in the institutions, and the infants were very young, they were too young to express any neurodevelopmental disorders they had. So we really think there is no reason why those who were adopted were more at risk genetically of any neurodevelopmental disorders. Okay. The study was started by Professor Sir Mike Rutter, who was the first ever child psychiatrist in Europe, maybe the world, but I don't know. And there are various investigators who have been involved over the years as well. And I'll thank people along the way as well. But let me tell you some overall findings from the study before I show you the brain imaging. So if we look at something like head circumference, okay, this is them in the institutions, this is them in the UK, and this is a marker of the head circumference. And this is the normative value at 0. And these are standard deviation units. You can see that the heads of pretty much every child was smaller. That's this is the first astounding finding I'll show you. Okay, here's the average. So we think, of course, this was affected by those early experiences, and that's what this depicts, but we don't know. And they were followed up at different intervals as well. And as an overall group, what you can see already is that there is a degree of catch-up. You can see this profound reduction in head circumference is much less profound here as young adults. There's still many with smaller head circumferences than you expect for their age, but it is much reduced. And you can fit a lovely line to this type of plot. If you look at IQ as well as our indicator of cognitive ability, again, on average, we see a remarkable degree of catch-up, which is quite astounding. Unfortunately, again, not every child has the same experience. Okay, but there is some good news. So, how can we dig into this a bit more? So, we've done various analyses which have shown us that if we look at those who were adopted before 6 months, on average, they have quite a different outcome than those who adopted when they were older than 6 months, which correlates roughly with about 6 months in the institution. Okay, because they entered at slightly various ages, but very early in life, typically. And these are summarized in this article here in the Lancet from a couple of years ago now. And these are the different follow-up time windows. I haven't got the scale at the bottom on this particular plot, but this is in about entry age 6-ish. This is age 11. This is age 15 to 16, and this is mid to late 20s. Okay. And you can see that we've also got a control group who are UK adoptees, and the light blue shows the Romanian adoptees from less than 6 months, and the red shows the adoptees from more than 6 months. And these are people that cross the threshold in terms of symptoms to be at risk of autism spectrum disorder. And it's very clear that those who were there for longer have a much higher risk, whereas those who were adopted before they were 6 months old have a risk which is fairly similar to what we'd expect in the general adopted population in the UK. Okay, which is actually not dissimilar from the general population. In terms of disinhibited social engagements, which is exactly what it is, it's I suppose related to attachment, but this is more based on the behaviour. The socially disengaged, you can see this is quite high in everyone when they come in, but in the group who are adopted before 6 months, This goes down to fairly normal levels, which stays high for those who are institutionalized for longer. This is IQ, cognitive impairment. You can see very high levels of impairment, but the levels of impairment really drop down into adulthood. Okay, there are still some individuals who are impaired, but you can see how this catch-up manifests very nicely here. And ADHD symptoms, overactivity, and again, we see the same pattern here, with this more than 6 month group showing higher levels. And then there's a bit of a spike in the tail because emotional problems, either reported by parents or the self, seem to emerge in early adulthood. These are depression and anxiety symptoms. And while they don't show excessive levels, there are some with difficulties. They don't show excessive levels in their teenage years. They really go up in adulthood. So we want to know what's happening in the brain. So based on the literature, we have an idea of different networks being involved. Oops. I think I mean to have the second slide. Yes, let's jump to this one. Since we planned the study, there's more literature, we added a network. And we think that various networks are involved because of their normal function and their association with previous studies. So we think the frontostriatal circuits will be involved, and they might be related to executive control. The ventral frontal circuits with reward processing, which might affect anhedonia, mood. Disrupted emotional processing, which will affect social engagement as well as mood symptoms. And there's a lot of emotional processing deficits in ADHD and autism as well. And deficits in what we call the default mode network, which is about understanding your internal states and switching between external and internal states, very important for attention and referencing the self and integrating, internalizing information, internal information as well. Impaired in most psychiatric and neurological disorders as well. I won't go through these networks one by one, but I just wanted to show you what our hypotheses were. We've done some structural imaging and some functional imaging. I'll focus on the structural imaging today. Just need to move on. So in terms of the study, we did look at brain structure. That's what I'll show you today, the results. We looked at brain function with functional MRI. I won't be showing you these results today, though it's still in analysis. I'll show you some of the neuropsychology that's published now. And we also did some connectivity work. Again, this work is still in analysis, so I won't be showing you this today. I'll just be showing you this diagonal here today. Who took part? Here are the numbers of people. So we did long interviews, different assessments, parent interviews in as many people as possible as young adults. This is the English Romanian Adoptees young adult follow-up. And then from this group, we invited some into the brain imaging study, ADOPTEES. And you can see as a percentage of the total final sample that we examined. There's also a UK adoptee group here, so the numbers won't add up to 100. You can see that about a little bit less came into the brain imaging study than the adult follow-up across the board, as we'd expect. But we know from an analysis of the demographic factors that they're quite equivalent. We don't have a biased sample. It's not that the most unwell or the most well are in the brain imaging sample. And here are the IQs on average of the sample. And again, we don't think we have sampled a particularly impaired or particularly well group, because you can see the differences between IQ are very similar between the groups. So the young adult follow-up is the data I just showed you on the symptoms, and the brain imaging data I will show you now. So we know already that within the first year, and the first year of life in particular, but the first couple of years of life, there's a great expansion that extends from the neonatal period to the postnatal period of grey matter and white matter. And we know these volumes, this is a simple stat showing the volumes, pretty much doubling over this time. Okay. And we know a lot about this developmental process, and there are genetic and experiential factors that do matter here. So they're good candidates to be sensitive to what's happening. We took brain scans from each individual, a T1 weighted image, which has got nice contrast between grey, white matter, and CSF. We put it through a free surfer pipeline, which allows us to segment the grey, the white, and the CSF, extract the surface, and parcellate into different brain areas. What did we find? So if we look at total brain volume, the first finding we found was a really dramatic reduction in the overall brain volume. So you remember that head circumference finding that I showed you, suggesting that there was some catch up in head circumference. The underlying brain volume is definitely lower in this group. You can see a nice range here. And one of the factors that explains quite a lot of variance in this range is the time spent in institutions, which is the x-axis here. This is going from 1 to about 40 months. And you can see the degree of brain volume reduction is related to time spent in institutions. So it's a very clear relationship. The longer time you spend there, the more likely is your brain growth will be reduced. When we looked at the birth weight when they came into the country, that gives you an index of their development in Romania. Looking at subnutrition, again we have limited data, but we have some data, and height, because overall these individuals are smaller. None of those explained this effect or this relationship here. Okay. So it is about the time spent in institutions, not about malnutrition. There were regional effects as well. We see this area in the inferior frontal gyrus was reduced. I mean, some of these measures of surface area, thickness, and volume that I won't go into today. And I'll show you some neuropsych as well later, but this relates to some inhibitory functions that we measured. We also showed the inferior temporal lobe to be different, but this time it was a bit bigger than expected after controlling, of course, for the total brain volume reduction. And then we show this area here in the medial prefrontal cortex. It was neither bigger nor smaller, but the actual surface area and volume here were related to the time spent in institutions. So we have a global effect, and we have 3 regional effects. And these also related to similar to memory impairments that we have. Just showing you this in a bit more detail. Here are these inferior frontal regions, and this is showing the relationship with time spent in institutions. So they weren't significant, but actually this area here in the inferior frontal cortex showed a slightly weaker but significant relationship with the time spent in institutions as well. And this is the relative size here. So this area is smaller, but when we control for the total brain volume, we see this positive correlation here. We also see, this is just to dig a bit more in detail, we also see this significant positive correlation here. This is what we expect from the control group. You can see this line here. So in fact, it's interesting, maybe this area reflects the degree of compensation. This might represent an area of real plasticity that can adapt. Of course, we need longitudinal studies to know for sure, but it does present that intriguing possibility that these areas might adapt. We're very interested in these subcortical structures. This has consistently been shown to be associated with negative experiences when, for example, those that lead to depression or PTSD, like abuse, for example, and maltreatment, particularly in older childhood. This is obviously very young childhood. But in our group, the subcortical structures that come up again and again, the hippocampus and the amygdala, showed no clear differences in their volumes and no clear relationship with the time spent in institutions. So this is a real deviation from other forms of maltreatment and negative experiences. So in terms of neuropsychology, we did a prospective memory task. This is a task where you have to remember future actions. So for example, we might say, oh, we're going to do some tests. When you finish the second test, can you please give me the pen back, for example? Or when you finish, I'm going to set this alarm. When this alarm goes off, can you please write down your thoughts on this piece of paper, for example? So you give people various tasks to complete during the testing session, and that's prospective memory. So, for example, if you go off to work and you realise you're low on something in the house, and you think on the way home I'll pass the shop and I'll pick this up, and then you walk past the shop and you completely forget, that's a failure of prospective memory. Okay, a memory for future actions. And that could be for many reasons. You might be easily distracted, or you might have poor encoding of those memories. Okay, and there are, I won't go into the model, but there's a model of prospective memory which is related to encoding, monitoring, maintaining the memory, and also obviously there's a retrieval function as well, which is related to spontaneous thoughts and intentionality. And we know that there are various brain systems that have been consistently involved in prospective memory as well. And here's an example of the test we actually used, showing like when I hand you a red pen, sign your name on the paper, and this is when the actions occur. And we just score people based on whether they do the tasks, and whether they do them on time, and they get points. Okay. So these groups in yellow and orange are our Romanian adoptees, and these are our control groups. We also have a healthy control group on these graphs. And you can see here that there is impairment in prospective memory, some risk-taking behavior, some emotional recognition, and also some proactive inhibition. When we control for the IQ differences, these two effects here remain significant. So we know there are specific impairments. So I'm just going to come to the end now. I've got a couple of summaries in the concluding slide. So the Romanian adoptees with a history of institutional deprivation, they show impairments across a range of domains. So I showed you the ADHD symptoms, the autism symptoms, disinhibited social engagement, IQ, and emotional symptoms as well. There is more impairment in young adult life in the Romanian groups compared to the control groups, particularly in those that had longer periods of deprivation. There's also, I didn't show you this, but there's more clinical service use in this group as well. But we have to remember that many of the adoptees were quite similar to the control groups, particularly those with 6 months or less deprivation. So I think one important message I want to give you today is it's not inevitable that if you've experienced deprivation, that will be a barrier to normal development. That is not what we are finding. There are clear impairments, but there are also some really good news stories from this group. We know there's cognitive impairment in this group, and again, there is a risk factor here from institutional deprivation. Some cognitive impairments are stronger in those that experience more deprivation. Some are there across the board, like prospective memory. And we also know that there is brain impairment in this group from the structural imaging I showed you today. And to conclude, particular brain imaging, we think that the current findings are consistent with the hypothesis that very time-limited events, negative events, that was experienced in the first few years of life can have an enduring effect on the brain because we can pick up differences in adulthood. They're still observable. What I didn't show you was the fact that the deprivation predicted later IQ and ADHD, so later ADHD, and the effects were mediated by the brain volume changes and the cognitive changes. So this raises the possibility that regional, there's also the effects actually in the temporal lobes and this medial frontal region, where the areas seem to be relatively bigger and not everything was smaller. So there is this possibility we might have some compensatory effects which protect children who've had these negative experiences from developing other disorders, as well as some other brain areas which seem to be mediating the risk for those disorders. I hope that's clear. There's always a lot of people to thank. The study is overall led currently by Professor Edmund Sanuga-Bart, my close colleague, and the imaging was conducted by our student Nuria Makaz and Dennis in collaboration with Graham, and everyone else on the list was involved in the other parts of the study. Thank you for your attention. I'll stop sharing my screen now. Hello again.
Yeah, hello. Thank you, Dr. Mehta. I must say the talk was really exemplary, and it was interesting to know how early childhood deprivation is associated with alterations, in adult brain structure and neuropsychological functioning. From your talk, we could imply that the changes in brain structure could play a somewhat a mediating role between the experience of, deprivation and the levels of cognitive performance as well as the mental health. With that,
Yeah,
I would like to take the questions from the audience.
sure.
So, one of the question asked is, what are some of the the structural abnormalities which you could predict in the brain due to childhood deprivation?
So some of the structural abnormalities that we predict in childhood deprivation, do I understand correctly the question?
Yeah, yeah, yes, yes.
Yeah. So based on the behavior and the cognitive profile of individuals, we actually predicted frontal and subcortical structures will be very important. So what we found were these cortical structures. What we didn't find was some of these subcortical structures. So some of our predictions were right, and some of them were not right. Now, if you look at other forms of maltreatment, like abuse, for example, what is very interesting is that you do see these subcortical changes, but it's not entirely clear how much of those changes are due to shared genetics with the environment, your parents, for example, and their genetic risk factors as well. And what's interesting is that there have been studies recently, I'm sort of going beyond the question, I'm sorry, studies recently looking at heritability and genetics around heritability. And I think this is to emphasize the strength of the study, is that, let's see if I can remember these studies correctly, but did I make some notes? Is that actually the genetics around, say, risk factors for abuse and consequences of childhood maltreatment, some of them do overlap quite strongly with the risk factors for mood disorders. So while there might be unique effects, what is not entirely clear is the degree to which the reporting of retrospective abuse is coloured by the current psychopathology. So in those big genetic studies, the people were asked, did you experience negative experiences in your childhood? Did you have negative experiences? Do you have current mood disorders? And it seems like those people who have current disorders and problems are more likely to say that they had experiences that were negative in childhood. But when you follow people up prospectively, that's not always the case. So these studies are very difficult, and that's why this study is very important, because we follow them up from the point they were brought into the UK. So we knew exactly what their experiences were. They're not chosen based on those who've got problems. They're chosen based on who's got their experiences. And so our brain imaging abnormalities that we see, we're thinking, well, this is quite extreme what they've experienced. So really, if you've got brain imaging abnormalities which are much bigger than this, we have to look upon that with caution in other studies. Yeah.
Thank you. Okay, so there is another question, like if you want to sum up that, how does early experience shape individual development? Just in, I know the talk was entirely on that, but if you just want to sum it up in, 1 to 2 lines, so how does early experience shape individual development?
So how does early experience, I missed one word in there. Oh, shape. Did you say shape?
No, just we all have our own development. We all have a childhood. So maybe just the early experience. How does the individual development? How does that influence or experience the early experience? How does it influence the individual development?
Of course. Yes, that's a very good question. So it's a real challenge for us to understand what we call normative experiences. So the variation in normative experience that we have, and some of those are good and some of them are not so good. Like if you say you're like a child and your parents, split up and divorced, that's a really terrible experience. We know that those experiences of living in a difficult household and that variation does have experiences on later psychopathology, your own mental health, mental well-being. We don't know very much about how that is manifest in the brain and its brain development. Those studies have not really been done very much. There are some examples, So we do know that there are effects, but there aren't that many studies as well. But the degree to which our normal variation of experiences that we have that are not classified as highly negative, that is not well understood. No. So I can't answer that question yet. We don't really know about the variation in normal experiences because it's very hard to measure this, and you need longitudinal studies to do so. So I think in the next 10 years, there are longitudinal studies that will start to report on this.
There's no problem.
In a way, the way I understand the question is asking about variation in the control groups and if we can understand that. And we don't have enough power in those groups to really answer that question. Yeah.
Okay. So, what would be the effect of, we have the twins. So,
Yeah.
like if we'll expose them to two different. Okay, there is some network problem. Yeah.
Oh, yeah.
So, like in case of twins.
Yeah.
You hear my question?
Yes, I can hear you
So,
now.
in case of twins, so do you feel that the early experience will play a role in influencing their development? If we expose, twins, we know that the genetic development almost the same. So, what kind of environmental influence do you feel?
Yeah.
Yeah.
Yeah. Well, there are studies which can answer that question much better than my immediate knowledge. So we do know a lot about this. There is a large study called the Twins Early Development Study, actually, which runs in my institution by close colleagues of mine. And they've been doing a lot of work on the genetics and the environmental risk factors that will affect development. And actually, we do know a lot about this. They've got dizygotic twins and monozygotic twins in the study, of course, so we can understand the role. And of course, there's a lot of heritability. If you look at mood disorders like anxiety, developmental disorders, there's an awful lot of heritability in these disorders. But I can refer you to those studies if you want to dig in. It's probably beyond, that's a separate talk, really. If you want to dig in and understand more about what those factors are and how they play out over the years, because that study's been going for a few decades now. And within that study, there are people that did experience early abuse, neglect, and maltreatment. And we know a lot about the risk factors for, say, PTSD in these groups as well. So there is a lot of knowledge being gained, but none of that group have been scanned. So we don't know about what's happening in the brain and what the mediating factors are. So I refer you to the TED study to find out more detail. So it's a bit beyond this short format, but I can tell you that we don't know much about the brain mediating mechanisms, but we do know that they're worth studying.
Okay, so there is a question from Dr. Nisha Patra, ma'am. What are the reasons for smaller brain volumes in your study?
Yeah, very good. Yeah, so I wish I knew the answer to that. So we know, so, what is the reason? So our first thought was maybe that they're just smaller overall. Okay, they've got a stunted development because they've been malnourished. That might explain it. So when we incorporate height into our models, the brains are still smaller. When we incorporate our assessments of malnutrition, so say the weight at entry into the UK, the brains are still smaller. So the idea that this is a very physical event related to poor development and poor growth because of malnutrition does not play out. So what's left? What the social and cognitive is left. So we think that this early experiences, we call it an experience expectant development. In our early years, we expect certain experiences, and they feed a very plastic brain, and they underpin its growth. And a lot of that development, a lot of that experience we have is social, okay, and it's based on cognitive development. So the model, I think, that I would say, I would guess, is dominating to answer your question is actually the social and cognitive experiences of very early age are very important in defining your brain development trajectory. And because that was lacking in this group, they're on a different trajectory, and one of the most obvious outcomes is the smaller brains. Oh, I've lost you. Oh, let's see what's happened. I think network problems, maybe. I don't know if other people can hear me, but I can see the comments as well. I don't know if other people can hear me. Yeah. I'll carry on, assuming that you can. I've got a question from Rakesh. Kids are getting stressed due to absence of classroom teaching in school environment due to COVID-19. Could you use those methods to handle their emotions properly? Oh, a slightly different topic to this talk as well. So I think it's slightly tangential to what I'm talking about. So I'll dodge that question. I'll skip it. I think that's a different discussion, but it's not that it's not important. I can put you in touch with some colleagues who are doing a lot of work in this area to try and understand the mental well-being of children. And if you look up Edmund Sanuga-Bark, who's the PI of the study, he's created a series of videos which are on our website to help guide parents in particular in helping handle this crisis, particularly with kids at home a lot. Sir, can you please explain how malnutrition specifically affects brain growth? Does early childhood deprivation affect regions of brain differently? Yeah, that's also a good question. Oh, you're coming back. I'll carry on until you're back. I think I partly answered that question in the one that before about the reasons for smaller brain volume. So what's interesting about the malnutrition is that we thought this would be an important factor. Over the studies of, we did early studies of brain head circumference, and now of course we've got the brain volumes themselves from the imaging. And really the malnutrition is a factor, but it doesn't explain much of the variance. And we think that this timing of the brain development is very, very important, the time window that they're in. Yeah. Of course, it's not that malnutrition will not have an effect. I don't want to send that message. Of course it will have an effect. But it doesn't seem to be a simple explanation of the brain volume differences. Obesity, yeah, very, very good. So I do have some other slides on this. I could have a look. But we don't see, I don't think we see very many cases of eating disorders in this group, actually. We don't see very many cases of psychosis, and we don't see many cases of eating disorder. So in this very extreme institutional deprivation, I don't think we can speak to this, but our data does suggest that childhood obesity is not a big factor here. And experimentally, how can we find this? I think we need much larger studies, actually, and we need to follow normatively. Oh, you're back. Hello. I've carried on answering the question.
Yeah, there was some internet connection. Sorry.
Fine. I'm quite happy. It's okay.
Could you hear me?
I can hear you fine. Can you hear me? Can you hear me?
Professor Mehta, could you hear me?
Yes.
Okay. So there is a question from Dr. Rakesh Pandey. Kids are getting stressed due to absence of classroom teaching and school enrollment.
Sorry, I answered that question while you were missing. I already answered that question. I don't know if the audience heard.
Okay, you have already answered the question.
Yeah,
okay,
that's all right. I can see them. I'm actually on a question from Jitendra, Dr. Sinha, and he was asking about experimentally showing the link between institutional deprivation and childhood obesity. Well, of course, it's very difficult to do experiments in children. Of course, this study comes close to a natural experiment. But I think what we need are larger samples and following children up with many different measures. And then, of course, looking at the groups in young childhood, older childhood, who are experiencing obesity to understand the factors which might explain this as well. And this is the best way. Thank you for the thank you. But biological or negative hormonal changes take place during brain development. Oh yeah, plenty. There are huge changes in hormonal levels during development. I mean, one thing I didn't talk about, of course, was puberty and the influence of puberty and the later pruning of the brains. And well, there's two things I'll say to that. The first thing is something I want to say in the talk, which I forgot, which is that why are the areas we're seeing the ones that come out regionally? Well, those areas are ones that show particularly rapid growth in those early years. But the parietal lobe also shows the same sort of rapid growth, but we don't see changes there. So while the rapid growth of these areas might enhance their sensitivity to these early negative experiences, there might be other factors. Those other factors might be the later pruning that carries on in later childhood and adolescence, of course, and the hormonal influences that go with that. So we know that cortisol levels do vary a lot in childhood, and we know that in these groups of adoptees, the cortisol levels are altered because of the chronic stress they experience, and that also can affect the brain development. Yeah. Another question. The brain is almost matured at 18 years. Why is the teen's brain different than 25? Well, that's very good. So if you look at the trajectories of grey matter and white matter, the peaks, if you like, on the curves vary from region to region, but largely they're in the 20s actually. So there are a lot of changes that take place. And if you think about, I'm going to be a bit more cognitive now, but if you think about brain development and growth, well, you've got this system, our brain, developing and growing, and it might reach some level of maturity by 18 years, but that's the point at which you can really start to still build and learn. So if you think of the brain as a structure, it's largely complete at 18. In fact, the brain during before teenage years, cognitively, is very similar to an adult, just in basic cognitive functions. There's still a lot of learning that goes on as well, and a lot of plasticity is still available. So there's a lot that can happen, and these transitional years from late adolescence to early adulthood, I think, are increasingly understood as more important, because about I think somewhere between a third and a half of all psychiatric diagnoses are made by the time they're 24, 25 years old. So that tells you that these early adult years are very, very important, and there's still a lot of changes that happen. Yeah. Great. So I think that's all the questions I can see. Dr. Swain, are you still there? Can you hear me?
Okay. Actually, sorry for the disturbance today. There is a lot of internet issue. Actually, there is some weather problems here. That's the reason we are having some internet issues.
Okay. Don't worry.
Anyways, thank you for handling the questions. It was really nice of you. Now I will request our Vice Chancellor, Professor Ishant Patra, to kindly address the session.
Thank you.
Okay, namaskar. Good evening. It was indeed a very interesting lecture. Are you able to listen to me, Mithul?
I can hear you very clearly. Thank you. Yes.
Yeah. This is more so because this is an area in which we work. We work in relation to maternal protein deprivation and early changes in the brain and how it would express in adulthood and during senility. So this is a subject which is quite close to my interest. So
of course,
many people enjoyed it. It was indeed a very interesting lecture. Like you were very clear in telling how early deprivations in terms of parental care or in terms of nutrition and all are going to really affect the brain at adulthood, particularly in relation to the environmental changes and the changes in the family structure, or adapted or non-adapted thing. I'm very pleased that you agreed to really address and come and join us in this venture of telling people about what is happening in various issues of mental health and health in general, and the current concepts and concerns. Um, I have one small observation I must make, that anything that happens in the very early life of an individual is going to really reflect it longer till death, and more so during senescence and during old age. So one should be very carefully handling the children that you have got.
Yeah,
I agree that in a joint family, there's a more enriched environment where the children grow better. But when they're only parents, two people working, they don't have time or they do not attend the children properly, do really the children suffers a lot. So the question is to have a child or not to have a child. If you do not have time, don't have a child. I always say like that. If you don't care, you cannot care a child, don't have one. It's not a pet.
yes, that's true. But I think also, I think we have to remember that what our studies also show is that there are some people who are very resilient, and we can't necessarily predict the development of a child into an adult, and there are so many factors. And one of the things that really surprised us was how well some of these children were doing as adolescents and adults, particularly those who were adopted early. That they can have some really negative experiences, but they can do quite well. But even those who were adopted later, some of those are still doing very well. So this is the next challenge. How do we understand success in growth despite your environment? I think
This is about studied people, children who have parents but stayed away from parents.
the children. Yes, maybe that's
They
true.
know their parents,
Yeah,
but still they stayed away from parents. How
exactly.
do they develop?
Yeah, I mean these are all questions that are remaining. I think there's an awful lot left to understand, and I think what our studies show is that while there are really negative consequences, there are some good outcomes. And even so, we have to remember that children are amazing, and our brains are amazing, and that some children are going to be all right, whatever the world throws at them, and some people are going to struggle, and we have to be careful not to judge because there's an awful lot of variability. Yeah.
Good. Thank you very much, Mitul, for sparing your time. Thank you. Thank you so much.
Thank you.
Formal vote of thanks from our team of organizers.
Oh, it's a real pleasure. It's a real pleasure. I'm delighted.
Yeah. Nilipta, take over.
And I'm happy to come back and speak again. Okay. Bye, everyone. Oh, sorry. I'm happy to come back and speak again if ever the opportunity arises.
So we have come to the end of this 11 days webinar series. On behalf of the organizing committee, I would like to propose a common vote of thanks first of all to the today's speaker, Professor Mitul Mehta, for talking on a topic completely, new topic in this webinar series. It was really interesting to listen to him, to you, Dr. Mehta. I would also like to put forth my gratitude to Professor Ishan Patra, Honorable Vice Chancellor, Ravenshaw University, for conceiving the idea for enlightenment, not only for this webinar series, but also for all the Ravenshaw 150 lecture series. Sincere gratitude to also all the 11 distinguished speakers who enriched us with their elaborative lectures. It was our privilege to have you all with us in this 11 days webinar series. I would like to acknowledge the support and contribution of Professor Meena Samanta and Dr. Unnishi Vedamata for all their support and for all their support. I would also like to thank you all others whose names I forgot to mention here, but who have helped us in all the logistics. Lastly, my heartiest thank you to all the viewers who choose to be live with us. The support and participation is surely encouraging for all of us for all the future webinars. Thank you.